Great, thank you very much. Nice to have this opportunity. My name is Ed Gillen. I'm the CEO of Grip Molecular. We're intent on transforming medical diagnostics. I think I can say safely that all of us have experienced to some degree the current state-of-the-art medical diagnostic processes and the difficulties associated with that. And in summary, it's too difficult to provide samples to be tested, and it takes too long to get a result. The reason that we as a global community have been struggling with medical diagnostics is that all of the technologies today are chemistry-based and require a trade-off between accuracy and speed. We're taking a completely different approach. We're using solid-state electronics, which give us the ability to provide highly accurate results in literally minutes and offers them at the point of need. The product is, as you see pictured here. The way that it would work in a home environment, for example, is that you would procure the device at a retail pharmacy for about $40. You would load an app onto a cell phone or any other medical device. And when that device is brought into proximity with the cell phone, it would display the results on the app. It provides a very easy to use, very fast result for multiple diseases, and simultaneously also provides the opportunity to assess the host response to those same diseases. It obviously facilitates telehealth interaction as well. We intend to provide this diagnostic information in the home, in elder care clinics, in retail pharmacy, and in hospitals and clinics as well. The first product on the technology will be an upper respiratory infection panel. There are 90 million people that need this test on an annual basis in developed world markets. If you multiply that by the $40 I mentioned earlier, it amounts to a $3.6 billion annual revenue generation opportunity. We've demonstrated proof of concept. We have issued patents. Development is underway in our labs, and we anticipate launch in 2023. We are currently raising funds via a convertible note. And have made great progress in raising those funds. We're using them to validate the sensor performance, and we do have interest in our Series A round. And with that, I'll stop.
Thank you, Edward. Judges, could I invite you to? Thank you.
Can I ask just a clarification? Is this a detection of proteins or nucleic acid, and is there any amplification steps?
So it's the detection of both simultaneously, and there is no amplification that's required. It's direct detection.
Okay.
So from a commercial model, why did you all decide to go direct as opposed to maybe thinking about licensing with multiple companies?
The challenge with licensing to companies is that as soon as you license the technology to one company, then that creates challenges to license that same technology to others. So we think that that would dramatically limit the application of the technology. We would be open to licensing the technology for certain fields to certain companies. That might be an option. But you also probably can appreciate that it also limits your exit opportunities, right? Because a company that wants to buy this, for example, would probably want to buy it all and not have it encumbered with other licenses to potential other competitors. So those are some of the reasons that we're not planning to take that course.
Thanks, Edward. This is Anna. I'm curious, the next pandemic, does this enable faster development of new tests for different viruses? And can you speak to that a little bit?
Yeah, so the biosensor can be very rapidly modified to detect new pathogens. So we estimate that from the time for example, the nucleic acid sequence was determined, 8 weeks later, we could be producing a product to detect that particular pathogen at high volume and at low cost. That would be particularly interesting, we think, also for influenza strains, right? So when you have the shift and drift phenomenon, we would be able to detect those specific strains and modify the device to do so within about 8 weeks.
Did I understand that you can detect both protein and nucleic acids and get a readout in such a short time?
Yes, sir, in a minute.
Has this been published?
Yes, there has been some publications on the base technology, and we can share those for those that are interested. And then, obviously we're moving forward with our research work, which we also intend to publish. And so there'll be additional publications that are coming.
And is it patent protected?
It is. We have exclusive license from the University of Minnesota where the technology was invented. And so we have access to those 3 patent families. There's another one that's in negotiations today. We actually have today 3 issued patents. 2 additional patents will issue this month. And then we have 7 others that are patent pending.
And I guess on the published works, is there a data that demonstrates level of sensitivity of this compared to traditional assays?
Yes, we have already gotten to picomolar sensitivity, and we expect the femtomolar will be achieving it in the next month or so. Obviously, that would be the level of detection that would be required for all the pathogens that we know about today.
Thank you.
So how much are you going to charge per test, and how much does it cost you to actually perform the test or come up with results?
Yes, so the average selling price would be $40. Our cost of goods in the first year of launch would be $12, but because of the amortization of intangibles, it drops down quite dramatically. So it's about $7 going down to $5.
I mean, it seems like your market is whatever you're charging times 100 million a year.
That's correct. That's correct. That's the $3.6 billion that I mentioned earlier. So it's a very, very large commercial opportunity.
Sorry to interrupt, but we're out of time. Thank you, Edward. Thank you, judges.
Thank you all. Appreciate the opportunity.